So we've talked about action potential, but what actually gets this whole process started? Turns out that there's these ligand gates, sodium channels that basically start that wave, basically getting that positive charge over to the other side of the membrane. And it's done through these gated ligand channels. Remember, ligand is something that has to activate it to get it started. So we'll talk a little more about that soon. Then there's voltage-gated sodium channels. And voltage-gated ones are the ones that will go ahead and carry that charge from one axon all the way to the end of it, and then pass it through a neurotransmitter. Also, when we go ahead and start that electrical energy, that depolarization event, it has to hit a certain threshold. And if that threshold isn't met, we go back to resting potential. And this happens quite a bit. We have to get past that crucial membrane potential, which is somewhere around 40 millivolts, to actually get the process started. So it's the minimum amount of current that moves from one side to the other. It comes across through these voltage-gated channels, and it brings those ions from one side of the cell to the other to get this process started. It's kind of an all or none principle, where once that electrical, so here's the axon, here's that wave of electrical energy, and you'll see that everywhere else it's pretty much positive, except for where that current is being changed or moved. As that current moves down that axon, you'll see that the wave is moving as well, so everything returns back or polarizes again, except for where that event is occurring. So what's happening is this wave is moving from one side of the axon, eventually all the way to the other side. It's doing it in a small wave right now, and it's doing it wherever that wave is. That's the only place that we see this depolarization event occurring. Everywhere else, it's polarized. So once we get past that refractory threshold period, we're going to go ahead and push that stimuli over to our next area of our axon. Then we're going to go to that hyperdepolarization. The cell can't be stimulated again, but it takes in some stimuli to get it back to resting membrane potential. And here are those channels that we talked about. There's different types of channels, but let's talk about the first one. The first one is a voltage-gated ion. I'm going to go back and show you a quick picture and come right back here. So you'll notice that with the axon, you'll see that this volt is basically moving down the axon. When this electrical energy comes close, we're going to see that these voltage-gated channels will go ahead and go from the closed state to the open state. So these voltage-gated channels have three states. The closed, that's the one that's going to be just where the cell is polarized. Ions can't pass through, and we just have sodium on one side, potassium on the other. We're really only going to concentrate on sodium right now because this is a gate that only one ion can go through, and that's going to be sodium. When voltage hits a certain potential, what's going to happen is, see these little areas of the protein? They're going to move out of the way. And by moving out of the way, it's gone ahead and created an open gate. See, now the gate is open. So now ions can pass through, and they're going from a high concentration to a low concentration. So these guys are definitely rushing in. Eventually, that electrical stimuli will pass, and then when that happens is we're going to inactivate the channel, and we're going to do this by blocking that gate. So even though this is the closed state, this is the open state, we need to stop it pretty quickly. And how we do that is by having this little area of the protein block the channel. And by doing that, what's going to happen is it inactivates the gate. The gate is going to go back to its closed position and wait for an electrical stimuli again to open it up again. So as one area becomes depolarized, the voltage will move from one area to the next, remember like in that picture we saw, and that voltage change is going to be passed from area of the axon to the axon to the axon until eventually we get to the dendrites. So here we see the picture. We see the polarized area, the depolarized area, and as this moves, we're going to see that the action potential is in progress. This is where we're going to go back. This is in our hyperpolarization right now. And then these are all in our resting membrane potential. They can be excited, but they need this current to come through to excite them, so then they'll be in the action potential. So let's go ahead and review everything we've sort of learned. Cells are polarized, which means that they're charged. Resting membrane potential is maintained by what mechanism? If you remember back, there's two. It's the sodium-potassium pump and the potassium leaky channels. During the resting membrane potential, the inside of the cell membrane is more negative and the outside would be more positive, and it's done because of those proteins. Those gated receptors can be opened by a couple of things. One of them could be a ligand. A ligand is like a key that's going to open the gate, and those keys could be a hormone, a neurotransmitter. That neurotransmitter could be from one dendrite to one axon, or a chemical. And the stimuli is going to come as a charge, which is volt. The volt is another thing that can open those voltage-gated channels. That's going to be current. So stimuli of the neurons, what's going to trigger it is those neurotransmitters. Remember at the end of those axons, we're going to have these neurotransmitters. The electrical energy is going to come down. Eventually it's going to go to the end of the axons and release those neurotransmitters. Those neurotransmitters, which you're kind of seeing them right here, is going to be touching a dendrite of another axon. So here's the axon, here's the dendrites, and so this is one neuron, this is another neuron. Remember, this is the pre-synapse and this is the post-synapse. What's going to happen is these neurotransmitters are going to flood and basically go to the dendrites. The dendrite is where we're going to go ahead and start that electrical energy down the axon. So what triggers these things from doing some sort of motion or some sort of thing? It's going to be all the stimuli that we think about. Light, heat, touch, movement. These are all things that are going to cause those changes to occur to give us information to our central nervous system. And remember, those action potentials are going to be where we're going to see that depolarization event, repolarization event, hyperpolarization, and back to normal. The resting membrane potential. And remember, the resting membrane potential, a couple of things are happening. There's a positive charge on the outside, negative charge, which is maintained by proteins that are negatively charged. And then, of course, we have our 70 millivolts. That's what energy will be it. And then if there is some sort of stimuli that comes down, we're going to be able to go ahead and do that polarization and repolarization event. So how do we get back to resting membrane potential? So we have a sodium-potassium pump, and you can see it right here. And what happens is, for every 2 potassiums that come into this pump, 3 sodiums are pumped out. So 2 potassiums in, 3 out. So that eventually is going to make it so we'll definitely have a much more positive charge on the outside and less on one side. So that is going to be the trigger to keep our inner cell negative and our outer cell positive. Keep in mind the negative also comes from these negative proteins. The other thing that happens is potassium will leak to the other side of the cell, but it's drawn back, and it's drawn back into the cell because remember all those negative charges are in here? So it's definitely going to keep the potassium more concentrated inside the cell than outside the cell. Sodium channels do leak, but we basically go ahead and have some sodium comes in. And then as sodium comes in, we have this pump that'll basically take those sodiums and potassiums and push the sodium out and push it back in. At depolarization is where we have those gated sodium channels open. And because of that opening, usually it's going to be some sort of ligand, so that could be a neurotransmitter or a voltage change that gets the sodium to start coming inside the cell and start to leak in. At repolarization, what's going to happen is we're going to shut this sodium gate and this potassium gate will open. So now potassiums will go to the other side because remember how the potassiums were really attracted to the inside because of the negative charge? And now that the negative charge has become more positive, it's realized that there's a high concentration inside the cell and a low concentration outside the cell. So it wants to go to the other side and sort of get to equilibrium. Hyperpolarization is where that gated sodium channel becomes inactive. So this is definitely stuck. And the gated potassium channel slowly closes. Not all stimuli do an action potential. Action potential is caused by actions of a subthreshold. So anything below a certain threshold, like remember it has to hit above negative 30 millivolts to do something. After that, it'll do the action, but below that it won't. So if there's anything below that, we call it a subthreshold, and it basically makes it so that energy doesn't move from one side to the other. And then let's keep talking about local potentials in our next video.